Hello everyone, my name is Senga Edize and I'm studying biomedical laboratory science at Inesri Hengeri and my registration number is 23-19955. I have assignment of applied immunology and have questions which we are going to be discussing at this moment. The first question is question 21 and it is what is cell therapy and how does it work? By definition, cell therapy is the use of serum containing specific antibodies to treat infections or neutralize toxins. By here, we take serum from the people who have recently recovered from the disease or volunteers who have been immunized, and we use that serum to treat infections by neutralizing the toxins. By mechanisms, we take antibodies and bind to antigens and neutralize pathogens. Then it facilitates phagocytosis via opsonization or activates the complement system. We apply this cell therapy for the snakes bites, for the rabies and diphtheria. Hereby rabies is where the dog can bite the person. Then the next question is what are the risks of the cell therapy? As you have seen, cell therapy is used as the immunization. Then it must be having some risks. The first risk is about allergic reactions. Risk of anaphylaxis or serum sickness due to foreign antibodies. The foreign antibodies can come to the patient and cause allergic reactions. Then there is limited duration. It provides only short-term immunity. And there is infections. Most of the time, infections cannot be the risk, but sometimes real risk of transmitting infections from donor serum. By here, serum can come with the infections, and during the injection of that serum contain infection can cause risk to the receiver of that serum. Another one is antibody mediated enhancement. In some cases, antibodies must worsen the infection. Then the next question is question 23. How does adjuvants improve immune interactions with vaccines? Adjuvants are substances added to vaccines to enhance the body's immune system to respond to the provided antigens. Then they can enhance immune responses by activating innate immunity, where triggers pattern recognition receptors like Toll-like receptors, TLRS, on the receptors. Another method is enhancing antigen uptake, where it increases the efficiency of antigen processing and presentation. And another one is amplifying cytokine secretion, where it creates an inflammatory environment that promotes T and B cell activation. Then the other one is balanced immune system, where they can help delete the immune response towards a more effective type, whether it is humoral, which is antibody-based, or cellular, depending on the nature of the pathogen. The next question is question number 24. What is the role of memory cells in vaccine? First, efficacy. Memory cells play a crucial role in the efficacy of the vaccines by ensuring a rapid and robust immune response upon subsequent exposure to the pathogens. Then the role of memory cells in vaccine efficacy are long-term immunity. Those memory cells persist. Memory cells B and T cells, they persist in the body longer after the initial vaccination, and then they remember specific antigens introduced by the vaccine. By rapid response, when the body is exposed to the antigen, memory B cells, they quickly differentiate into plasma cells that produce large amount of antibodies, and T cells At the same time, they proliferate and mount on strong cellular immune response. Then, by protection of against variants flu, like influenza virus, sorry, the memory cells can recognize and respond to variants of the pathogens, providing broader protection, as I have said, to the influenza virus because it can vary. Those memory cells they can provide broader protection. The next question, which is the last question, is question number 25. How are anticellular produced and what are their applications? Anticellular production process by immunization. A host animal, example rabbit, horse, goat, mouse, is repeatedly immunized with a target antigen. We take the antigen and immunize those host animals. Then antibody production. On this process, the immune system of the animal generates polyclonal antibodies against the antigen. By here, as I'm saying, the first process of immunization, we take that host and inject the target antigen. Then that host itself produce the antibodies against the antigen on the second point. Then the third one, we correct the blood and serum is separated in that corrected blood. Then the last step is purification or optional, where antibodies may be purified using affinity chromatography for specific applications. Then by the applications, we apply them in diagnostic testings by using in immunoassays to detect antigens or pathogens. And the next one is passive immunity, where anticells provide immediate immunity against toxins or pathogens, examples snake antivenom. Then the last point is about the research. We use them to identify proteins or other molecules in experiments.